Good morning. My name is Rong. I'm a student in Scientific Computation Lab. My topic today is brain mapping and its labeling on the cortical surface. This is the outline of my talk. Since brain mapping is a new research area, it's understandable that some of you have never heard of it. So I will spend some time on talking about the motivation and its research areas in the computer science. But the main part of my research interest is in the labeling on the cortical surface of the brain mapping. I will talk about the previous work and the two major parts in the whole process. In conclusion and future work, I will comment on the limit of these methods and propose a future solution. Actually, everybody knows that no two people look exactly alike. So the same thing, no two human brains are exactly the same. But the classic atlas of the human brain derived from a simple brain or just from a very small number of subjects. So the result is a very rigid and sometimes inflexible system. So the useful information such as variability among the subjects are ignored. In order to change this situation, about 10 years ago, the International Consortium of Brain Mapping Group was formed by some scientists over the worldwide. The leading founder is Dr. Zhang from UCLA. So the goal of the ICBM is trying to create a probabilistic atlas of the human brain from a large sample of the people, say 7,000 subjects, and the people are chosen aged 18 to 90 years old. It's an adult, healthy human brain from a wide racial distribution. So Dr. Zhang, in his books, Brain Mapping Disorders, argued that with thousands of healthy brains, then the degrees of what constitutes the normal can be much more better understood. So with this theory foundation, we can say this brain mapping can provide approaches for your research in the brain disorders, for example, the Alzheimer's disease. Another thing that in the recent years we have witnessed a relatively exponential increase in the interests in the neuroimaging, and their product is huge. However, in the neuroscience community, scientists are frustrated because with the huge data, only a very small fraction are fully analyzed. So in order to improve this situation, we have this complete atlas to provide this framework in which we can input the ever-increasing data sets. This is a very important motivation. And the last one is, as we know, the relationship between the function and the structure of the human brain is very complex and poorly understood. So the future, actually the current project that they are doing is trying to set up a four-dimensional probabilistic atlas. Four dimension means three dimension in space and one dimension in the time. So it's not just simply a geometric pictures. It also superimpose associated attributes such as blood flow, receptor density, and etc. So with this completed atlas, the aggregate data in this anatomic atlas will give you very greater insights about the relationship between the structure and the functions. In my first presentation, I have given you a very simple scenario in the hospital setting in the future. So try to review a little bit. So this scenario, a patient has just a seizure. She goes to the doctor, but her MRI result and her 3D reconstruction map does not show anything wrong. So, but since we have that huge database with 7,000 healthy brain data, so we can compare them and And this database can give you the result and show you somewhere, for example, this folder might be the abnormal place. Since brain mapping is very complicated, so it's pretty much involves every research groups in computer science. For example, the computational biology will gives you the fundamental biological structures for the modeling, and the neuroimaging part is hard. So it gives you the implementation of that models. Also, the database and the network can advance the bioinformatic images for diagnosis and treatment. Of course, software engineering is very important. They can produce the software so the user can use the software to do the brain mapping automatically. And the last is knowledge management. So let's first take a closer look at the human's brain. Since my topic is about the extraction of the human cortical surface, so the major structure of the human brain is composed of patterns. It's just a very outstanding two parts. One is called Jiri, the other is called Suki. So it's very easy to see. In order to map this cortical surface, you have to find the boundary of this cortex. So that's our problem, and that's what we want to solve. In the application, all these fields, so for example, the neuroanatomic teaching and the surgical planning, or the sharing of clinical information, all rely on the segmentation and the labeling of this cortical surface. Actually, there are a huge number of approaches to deal with the human brain labeling and the feature recognition. Here, I just give a very short list of some typical approaches. They do it with the rigid models. So due to the time limit, I will just give you a general idea of rigid models. As the name implies, these rigid models cannot give you a very flexible atlas, so the variability among the subjects are not very well described. That's why these deformable models appeared. These deformable models is very good in the active searching this surface of the human brain. So this is a 2D example. I put it here trying to give you a basic concept of what deformable means. That means the atlas can keep deforming and until it shrink to the final shape of the human brain. So the scientists and researchers extend that concept to the 3D and apply it into their brain mapping research work. However, the drawback of this is it involves user intervention to find the matching Suki in order to map the human brain. So the last approach is the level set method. It's very popular for the last 5 years. However, this method also involves your user intervention to put the initialization and the fold annotation part depending on the resolution. So what we want? So we want our scheme, it's independent of the resolution. We also want it can describe the inter-subject variability. This is the pipeline I have talked about last time. So it's quite clear. It's from this workflow, its input is a stack of MRI data after some visualization tools, and finally we got this topological graph. With this graph, we can map the human brain. Last time, I think there are some confusions in the isosurface extraction, so I will spend more time on that part also in the segmentation. The input is this stack of MRI images. However, in order to build a 3D model, we have to let those images aligned. So if it is not aligned, we have to do some job. So this paper is a very decent approach to do the aligning of image data. Meanwhile, the images also contain a huge amount of noise. So we have to do the smoothing filter to denoise. Now we come to this, then after the pre-processing, we have come to the major part. It is called the isosurface extraction. In order to better understand the isosurface, I give you an example of the 2D part. In the 2D, these contour lines are also called isolines. For example, in this picture, these contour lines reflect the ISO value 5. So in 3D, a surface with the same ISO value is called the isosurface. Then extracting such an isosurface is called isosurface extraction. The volume data sets from CT and MRI, they contain 3D scalar field. 3D scalar function is a function of x, y, z, 3 variables. So the z variable, you may wonder what z stands for. So z variable means in the direction of the different images. So simply, the isosurface extraction is simply asking one question. Is the point of x, y, z inside or outside the isosurface? So in order to do that, we use the marching cubes algorithm. Marching cubes is a divide and conquer approach to locate the surface. They just subdivide the whole volume into units with 8 values at 8 corners. So if this value is bigger than the ISO value, we just define it's outside the isosurface. If it's smaller, then it's inside. So for example, we have this vertex V1 and V7 has values bigger than the isosurface. So these two vertices should be outside the isosurface. Then we create an index. This index corresponding to each bit. So the 1 means this vertex should be outside the isosurface. And after that, we calculate the contour location by some interpolation method. The final goal is just to create a triangle mesh with this information. So the triangle mesh can always give you a surface. For example, that 1,000 triangular mesh give you a very rough surface of rabbit. This is almost 70,000 triangle mesh, gives you the future surface. After that, after this cube is done, we can march to the next cube. That's where the marching cube name coming from. So the marching cubes for 3D isosurface generation gives you 256 ways for the surface to cut the cube. But by the symmetry, we can reduce it to the 14 tabs. Then after you understand that, then you will know that the isosurface value is very important, because with different isosurface value, we get different models. So this picture shows you with different iso values has a different surface. So in order to pick up the correct ISO value, we use these tools. This tool can give you a visual output of what the perfect cortex should be. So after that, then we have to do the denoising thing, because in the Z direction, there are still some noises existing. Another consequence is after the isosurface extraction, there are some isolated components generated. So this is also results from the noise which are not denoised in the previous part. So we use some algorithm to get rid of that parts. Then previously I have talked about this surface segmentation. It is curvature based, and it's based on the theory that every human brain cortex contains the elliptic behavior and the hyperbolic behavior. So to distinguish between the convex and the concave, we use this mean mean curvature operator And we still also need to distinguish the hyperbolic and elliptic. We use the Gaussian operator. Finally, we got that the segmentation result is divided the human brain into 4 distinct parts. And with that information, we generate the topological graphs. With that graphs, we can compare this atlas brain and the patient brain. So in conclusion, I have given you a general picture about what the brain mapping is and what we did to handle that. But there is still some limitation existing. For example, there are 2 3D models generated from the same brain but at a different time. However, we can see the obvious difference at that part. So we use this conformal mapping to handle this. That is our future work. Because, that leaves a huge long-run research work for you to do. This is a list of my references, and thank you for your attendance. Your questions are quite welcome.
When, first of all, these 7,000 brain database, is that already existing?
Now they have 5,600 is done. Their goal is 7,000 subjects.
If you have a scan that you do and you compare it to that database,
Mm-hmm.
You gave in the very early slides an example of the kinds of output, but where do the neurologists see the value here? Like, what is the reason for doing this? And I guess in clinical terms, what's it buying them?
Because the reason is that with this huge number of the human brain data, then you can see what's the normal range of a healthy human brain, right? So with this baseline, then you compare this patient with this baseline, then you know something might be go wrong. So actually, this whole project was proposed by the doctors and neuroscientists because they are frustrated with this. They cannot compare these things with. So what's the meaning? What's the difference, or what's the degree of the normal human brain can go?
How do they know what normal is?
So that's why they have to sample a huge data. Actually, they are
They
not.
can all be pathology, though.
Actually, they're not proposing that this method can give you explanation for the relationship between function and the structure. But with this aggregate data, they are hoping that they can be used for the future study. Yeah, as I know, so that's why I put 3 motivations there. It's I hope it's quite clear for you to see the whole picture. And
I always see the motivation of people collecting databases. What I wonder is if they've accomplished anything. In other words, if you compare a brain against this, unless you've categorized the data in the data set and know what normal or variance and so on are, you don't have anything. You just can compare and say it's like 1/3 of the brains or like 1/99th of the brains, but it doesn't tell you anything that I can see here in terms of providing a comparison. But maybe there's some analysis that goes on of the data in the data set.
Yes, because what I'm doing here is just for the visualization part. I didn't go what's the medical part, how the analysis is going on.
Okay, so they've done work on characterizing the different
characterizing
data sets that they've
analysis. Yes, they are doing that.
And is this entirely by MRI imaging, or is it by other types of imaging?
This project is for the MRI images, but they are dealing with function MRI also and the other new medical images. So, question there.
Also, this might help because one of my papers I want to present deals with 3D CT scans of brains, and what they do is even they use the atlas, but as they say, the original atlas, like it was just one brain of one individual, and it was not very useful. So if you sample it across large different ethnic groups, different populations, you're going to get a more realistic model. You cannot do what I'm going to talk about. It does not have an exact matching, but what happens is they can kind of do maybe allocation of a lesion based on maybe transposing this brain onto another one just to determine where the lesion is located approximately, as opposed to just, discovering pathology itself. As they usually use other methods to analyze the image itself, but when they're trying to find it in a 3D space, they use the atlas just to get, use the coordinate of the atlas to get the feel of
So it tells you on an average where certain anatomical parts of the brain are.
are going to be found. Exactly. So if you have a lesion, let's say you detected a lesion, then it can help you decide. Like if you didn't see it the first time around, it can help you locate it on the atlas. Of course, it's not going to be precise because no two brains are alike, but you can get some kind of idea of where it's going to be in the 3D.
Other comments or questions? Thank you very much.